Hello, everyone listening and watching. In this short tutorial, I'm going to give you a demo of how to use the interactive mapping module of the Broad Institute Genomics2Proteins Portal, which is a discovery platform to link genetic screening outputs to protein sequence and structure. So this particular module has two submodules, as you will see after we dive in. And this tutorial will focus on the first module, and then we will have another tutorial for the next one. So let's click on the Analyze Your Data button, which is the beginning of the workflow. So when you click there, the portal will prompt you to sign in using your Gmail address. So let's do that. And then it should show you a disclaimer. And as I continue, then we can see that there are two submodules within this Analyze Your Own Data, or what we say as interactive mapping module. So we will focus on this particular submodule here, which is how does the workflow look like if we start with the human gene or protein identifier. Clicking here, the workflow will launch, which involves this fourth step. In step one, you will select a gene or a protein identifier. From here, you can search up a gene name of interest or UniProt identifier. Here, I'm going to click on this example gene name CDKL5. Then you will see the FASTA sequence for the protein. We will proceed, and then we are now at step 2, which is the structure selection part of the workflow. Here you can select an experimental structure from protein data bank or AlphaFold predicted structure by both AlphaFold 2 and 3. So in this page, when you click on a structure, you will also be able to preview the structure. So in AlphaFold, if I now click here, then we have a preview of AlphaFold structure. I'll go back to Protein Data Bank, we'll select back the top structure, and we'll hit this next annotation step of the workflow. In this step now, if you come to the annotation viewer and click on this link, you will see the allowable formats in which you can input mutations or scores that can be numerical values and features as discrete or categorical values. And you can map these annotations to protein sequence and structure side by side. So let's close this and load some sample annotations given here, such as mutation and scores, and then features. One important note here is this particular column, the first one, residue position, is a mandatory column because we need this column to map the annotation on sequence and structure. And we can also combine all three types of data that you can map and paste the combined annotation in this form. So let's download a template which involves a combined set of annotation. From here, I'm going to copy these annotations and paste here. But note that you can also upload that file as CSV or Excel format and can do the same, execute the same workflow. So here now, if I submit, then it will give you this preview of the annotation table. Now each cell of this table is editable. For example, let's try editing this residue position to 13 here. And now if you hit submit annotation, it is going to show an error that you cannot upload a mutation at the position 13 for the residue position 12. So we will need to correct it, and we can also change this synonymous mutation lysine to lysine to arginine, for example. Another important note here is this category of annotation is editable from the column header. For example, if I now edit this category of data into mutation, then the portal will detect that this is an erroneous annotation because mutations cannot be quantitative values. So I will change it back to score as it should be and will submit the annotation. But before going there, you can also filter your columns. For example, if you just upload your data file with hundreds of columns and not all of them are necessary, then you can check off these boxes and you can filter the columns. And also we show a note that more than 30 columns may lead to performance issues. So we'll submit the annotation, and then now the annotations are mapped on the sequence space and also on the structure space. Now from here, there are a couple of options. For example, you can edit the color of the annotations. Here the mutations are mapped in red, but I can click on this color wheel and change it to a different color, blue. And you can change these colors for all the annotations. We can also click on this View Additional Annotations button, which brings in mutation data, variant data from Nomad, CleanVar, AGMD, so 3 different variant databases. And also it brings in a suite of protein sequence and structural features that we have available in the Genomics2Proteins portal, but in the resource module. So for example, for this particular gene, if you want to map also the available clinical pathogenic missense mutations in the context of your own annotation, you can select these mutations from here and can map those mutations. So now the orange spheres are the clinical mutations and these blue mutations are the mutations that you have uploaded. Now from here, you have multiple options to share the data and also share this workflow. So for the screen shot of the sequence viewer, you can click in this button and you can also download all this data by clicking here. You can download this mapping on the structure by clicking on the PyMol script button. But here I'm going to focus on how you can share this entire workflow to a collaborator, for example, and then the collaborators will be able to load this entire session in their computer. So let's hit share. And from here, I'm going to share this with one of my other personal email address. And I'll add it and then I'll create the link. So now this link will be accessible only to this email address. So I'll copy this and let's try to load it separately. So now you are given a prompt that you need to sign in using the authorized email address. So I will do that. And then we'll need to copy that link one more time. And then the same session that we created is now available to the collaborator. Remember that this session needs to be accessed within 7 days, and then we clear up the logs. So this was about the workflow for executing the interactive mapping of Genomics2Proteins portal, starting with the gene protein identifier. So stay tuned for the next tutorial where I'm going to show a demo for the next module, which is start with your own protein structure.